Welcome to the Basics lecture on biomolecules. We've already talked about how covalent bonding creates molecules via shared electrons. Carbon, because it has a bonding capacity of 4, is often the backbone of biological molecules. There are several common arrangements of biological molecules, and they include functional groups that are often found bound to a carbon molecule. You'll need to learn the 7 major functional groups and whether each is polar. See the figure in Chapter 4 in your book to fill out your notes. The first half of the figure covers hydroxyl groups, carbonyl groups, and carboxyl groups. The second half of the figure covers amino, sulfhydryl, phosphate, and methyl groups. Acids and bases are another common type of molecule. Acids tend to lose hydrogen ions or protons, same thing. The most common functional group to act like an acid is the carboxyl group, shown here. In this amino acid glutamate, the hydrogen easily falls off the carboxyl group and leaves the carboxyl group negatively charged, like this. Bases are molecules that tend to pick up hydrogen ions. The most common functional group to pick up a hydrogen is the amino group. In this amino acid lysine, the R group picks up a third hydrogen and becomes more positive. In biological systems, these charged forms are the ones seen more often. Once you've filled out the table in your notes, be sure to add these facts, and I'm expecting you to be able to determine which of the functional groups is polar. Now let's put this information together to look at 4 major biological macromolecules. All have carbon backbones, and 3 of them are considered polymers, which means they're long molecules built from repeating subunits. Lipids are not polymers. There are many different types, and they are made from different subunits. The forming of a biomolecule uses a dehydration reaction. The ends of the subunits of 2 parts of the molecule come together to form water, and the remaining carbons bind together, as shown here. In the reverse hydrolysis reaction, the carbon-carbon bond is broken, and the hydrogen and hydroxyl groups are attached from a water molecule to form 2 separate molecules. Both of these reactions require an enzyme to make them energetically favorable. Here's our first biomolecule. Carbohydrates are important for fuel and as structural molecules. The dehydration reaction that forms carbohydrates is called a glycosidic reaction. Lipids are a disorganized group that share the characteristics of being insoluble in water. They're either hydrophobic or amphipathic. Fats have a simple 3-carbon backbone called glycerol and bound to 3 long hydrocarbon fatty acid chains, shown here. Phospholipids have a similar backbone and 2 fatty acid chains. Fats are important in energy storage and membrane structure and are formed using an ester dehydration reaction. Another important lipid type are steroids. These molecules are amphipathic because they have a carbon ring structure and a small polar head. Here's an image that shows you several different steroids, from cholesterol, vitamin D, testosterone. You can see how similar they are. Cholesterol is the precursor to many other steroid molecules that the body makes. Proteins are a hugely interesting group of molecules that have dozens of different functions in the cell. So far you've seen them as pumps, microtubules, motors, receptors, and enzymes. A protein consists of a polymer of many amino acids in a particular order. We define a polypeptide as a single polymer, and a protein is made of 1 or more polypeptides with a specific shape. The structure of the amino acids themselves is very important. All amino acids share the same backbone, as shown here in purple. They all have an amino group on one end and a carboxyl group on the other end, and a hydrogen on the alpha carbon in the middle. Each of the 20 amino acids in biological systems have different R groups that are attached to the other bond on the alpha carbon. The difference R groups determines which amino acid it is. You don't need to memorize the different R groups for all 20 amino acids, but you do need to recognize whether an amino acid is polar or nonpolar. 5 of the polar amino acids are also ionic, where bases have gained a hydrogen ion or acids have lost one. Remember, you can tell if a molecule is polar by which functional groups it has. The dehydration reaction in proteins forms a peptide bond. There are 4 levels of protein structure. The primary structure is the amino acid sequence that we've seen already. There are 2 very common secondary structures that are caused by hydrogen bonds between the backbone molecules, shown here. In an alpha helix, a single coil of amino acids forms a coil, and the beta pleated sheet is an attraction between nearby strands, forming hydrogen bonds with each other. Tertiary structure is the interaction between R groups of amino acids that are near each other as the protein folds. The interactions can be strong ones, like ionic bonds between an acid and a base, or they can be weak ones, like hydrogen bonds. Sulfhydryl groups are also a form of covalent bond because cysteine amino acids, when near each other, can become oxidized, their hydrogens become removed, and these bonds are called disulfide bridges and are very strong because they're covalent. Lastly, parts of the protein that are nonpolar tend to find themselves on the inside of the protein, far away from water. Quaternary structure, the last level, is the aggregation of 2 or more polypeptides, and many proteins are made up of multiple polypeptides, like collagen or aquaporins. The final macromolecule is the nucleic acid group. Their main function is to encode and express genetic information, but they also form some cell structures, like ribosomes, and act as energy storage molecules, like ATP. The structure of polymers are the repeating monomers called nucleotides. Each one has a pentose sugar, one of 5 different nitrogen bases, and 1 or more phosphate groups. The 5 different nitrogenous bases are the source of genetic coding. You don't need to be able to draw them from memory, but you do need to know the difference between purines and pyrimidines. Notice that purines have 2 rings and pyrimidines all have 1. When a nucleic acid polymer is formed, the sugar group from one nucleotide is bound to the phosphate group of the next. This is called a phosphodiester dehydration reaction. When nucleic acids are double-stranded, the bases form hydrogen bonds to keep the 2 strands together. This completes our Lecture 8 Basics video. Write out the answers to these questions to study for the quiz.